there, in this video we are going to review our DNA notes. That is from packet page 52, and I have glued into my notebook on page 49. So to start out, DNA stands for deoxyribonucleic acid, right? DNA. DNA is a polymer, right? We know it's a macromolecule or biomolecule or organic molecule. So it's a polymer made of repeating monomers called nucleotides. And a nucleotide is a phosphate, sugar, and a base. And because this is DNA, the sugar is deoxyribose sugar, because we know sugars end in ose. Now for DNA, there are 4 different bases that can make up our nucleotide. There's adenine, thymine, cytosine, and guanine. Talking about the structure of DNA, DNA is made of 2 strands of nucleotides. Its shape is a double helix, right? Sometimes referred to as a twisted ladder. There is a base pair rule that governs which of our bases pair in order to form that double helix. So adenine always binds with thymine, and guanine binds with cytosine. This base pair rule is very important, and so one thing we can do to remember our base pair rule is that there is A and T University, and then we have Guilford College. So that is our base pair rule. These bases are held together with a hydrogen bond, and that is a weak bond, hydrogen bonds, but strong enough to hold our DNA together. So we have a picture of our double helix. So we're going to label the picture with hydrogen bond, sugar, nitrogen base, and phosphate. So we're going to start with phosphate. That's going to go right here. So phosphate, here's our sugar, and specifically, right, it's our deoxyribose sugar. Then we have our nitrogen base, which could be either adenine, thymine, cytosine, or guanine. And then we have a hydrogen bond holding those 2 bases together. So if this was adenine, then this base would have to be thymine. And if this was cytosine, this one would have to be guanine, so on and so forth. The sides of the DNA ladder are made of alternating phosphates and sugars, right? So if this kind of lightly colored part, so phosphate, sugar, phosphate, sugar, phosphate, sugar, so on and so forth. And then the rungs of our DNA ladder are made of the nitrogen bases, and they are held together by a hydrogen bond. Going back up to the top, talking about the location and function of DNA. DNA is located in the nucleus of eukaryotic cells. Remember, if we're talking about prokaryotes, that would be in the cytoplasm. DNA is genetic material that controls the production or synthesis of proteins. So location is in the nucleus, function is to have the instructions to make proteins. And we know proteins are very important because they create things like hemoglobin and insulin, which help us to live. We're going to do a quick overview of DNA replication in our DNA structure notes. We need to know that DNA replicates before cell division. Right? I'm sure you've heard of mitosis. So DNA has to replicate before cell division. The goal of DNA replication is to create an identical copy of the DNA molecule, right? That way each new cell that you make, whether it's a heart cell or a skin cell, has an identical copy of DNA. We don't want any mistakes. So the steps of DNA replication, step number 1, the DNA helix unzips and the hydrogen bonds are broken. So it's not a twisted ladder anymore, it'd be a flat ladder. And then we start to break those hydrogen bonds. Complementary nitrogen bases in the nucleus bond to the unzipped strands and 2 new DNA molecules are produced. In this process, each DNA molecule produced after replication has 1 strand from the old molecule and 1 new strand. So we say that DNA replication is semi-conservative, right? Because each new DNA has a copy of the original parent strand and has the new strand. So it would be like that. And we'll talk about more about DNA replication in the future, but these are the notes we need as far as the structure and function of DNA. If you have any questions about our DNA notes, please let me know.